DNA carries genetic material so living things can function. It is in a double helix shape, double-stranded, and is composed of nucleotides. DNA is in the nucleus of the cell. Nucleotides are the monomers of DNA. Nucleotides are composed of a nitrogenous base, a sugar called deoxyribose, and a phosphate group. The nitrogenous bases are A, adenine, T, thymine, C, cytosine, and G, guanine. Purines are a two-ring structure, and they will always pair with pyrimidines, a singular ring structure. The purines are adenine and guanine. The pyrimidines are thymine and cytosine. One purine is equal to its counterpart pyrimidine. The purine adenine will always pair with the pyrimidine thymine, and the purine guanine will always pair with the pyrimidine cytosine. A and T will always pair together, while C and G will always pair together.
So we can grow as we get older. Our cells need to replicate, which means our DNA needs to replicate. This can be done by a few enzymes: helicase, DNA polymerase, and ligase. Helicase is an enzyme that will break the hydrogen bonds that are holding together the nitrogenous base pairs. DNA polymerase takes free-floating nucleotides and adds them together to make the complementary strand. Ligase is an enzyme that will bond these nucleotides together and make sure everything is sealed. To speed up this process, helicase enzymes run in both directions. If they didn't, this process would simply just take too long.
Because DNA is composed of one old strand and one new strand, it is a semi-conservative model. DNA is anti-parallel, which means that one strand will run 5 to 3, while the complementary strand will run 3 to 5. DNA is always read from 5 to 3. When helicase unzips the DNA, it has a leading and lagging side. The leading strand is going in the same direction as helicase, making it the 5 to 3 strand. DNA polymerase is able to move with the helicase. The other strand is the lagging strand, because nucleotides are having to be read and added to the 3 to 5 end. This results in the DNA being added in fragments called Okazaki fragments. These fragments will then be glued together by ligase.